So before we move to close the voting, let's go ahead and open the questions from online.
So first one from Anesti Lazaridis. Has the company made a final decision on the cohort number required for the current trial, i.e. 10 or 15 participants?
So the answer to that is no, we haven't made a final determination. We designed the study to be open up to 30 patients to give ourselves the flexibility to continue to investigate as we saw fit. At this point in time, we believe that achieving somewhere between 10 and 15 patients will give us sufficient information to make a decision on it for going forward, but we don't yet know specifically whether 10 will be enough or 15 will be enough.
Thank you. Another question from Mr. Abhishek Prasad. What is the realistic timeline that we're looking at completing the trials? Also, does loosening of restrictions help with the trials?
So as I mentioned in the chair remarks, we are hopeful that the loosening of restrictions as we're coming out of the pandemic, so to speak, will in fact improve the ability for patients to access their caregivers, be screened for cancer, and potentially be available for the study. It's still very hard for us to predict the rate of recruitment at this stage. We have 4 sites that are all actively screening patients. We meet with them on a weekly basis to try to look at what their screening numbers look like. We are still hopeful that we can achieve meaningful numbers of study subjects during the course of 2022, but it's not something that we're really able to predict at this time.
Thank you. Further question, of the patients that have completed the trial, why can't the investigators or the board tell shareholders whether they can detect cancer?
Yeah, so this gets to that question associated with how many patients do you think you need to do this? We, at the interim results that we reported for the first 5 patients, what we are able to report is our observations from that, but we're not able to draw conclusions from an N of 5. And so what we're really, it would be imprudent of us to try to suggest whether we can determine effectiveness at 5 patients or at 6 patients or when we get 1 more patient. So what we really are trying to achieve is that we have enough evidence that it's pointing us in a direction with confidence. And today what we've been able to say is that we're very encouraged by what we see with the first 5 patients, but we can't make an individual call on any given 1 patient. And so it's not appropriate for us to report what we see on 1 patient versus another. We have to wait until we accumulate enough information to be able to say, is this looking like it's moving in the right direction that we can ultimately submit to the regulatory authorities to get clearance for its use as a clinical aid.
Yep, thank you. And another couple in relation to the trial. In light of the protracted phase 1 clinical trial, is the board and or management confident in our ability to expedite the anticipated follow-on pivotal trial?
So again, referring back to the remarks that I made, while we can't draw conclusions as to whether or not, answering that prior question, we can say we can detect the cancer or not. What we can say is that we're significantly encouraged by what we see in the study so far. And as a result of that, in fact, we are starting to make plans for the next phase of clinical involvement. We're encouraged enough that we believe that it's time to start to progress towards what the next phase would be, moving towards a pivotal study. And so I think that the short answer to that question, Mr. Hollis, is yes, in fact, we believe that we have enough evidence from what we're doing to A, continue the study here in Australia, and B, begin to make plans for the next phase of testing.
Yeah, I think this flows in. Mr. Robert Scanlon's asked a question online. What are the immediate next steps following a successful trial?
So immediate next steps would be to go beyond Australia, to move into jurisdictions where we can then look for US and or European regulatory authorization as well. So what we'll be doing then is making additional HER2 imaging agent to be used in larger studies that would be done both in Australia as well as elsewhere.
And another question from Mr. Scanlon: what are we still needing to learn from the current trials?
So probably, I think the shortest answer there is while we have evidence that the nanoparticles are reaching the lymph nodes, What we need to know is what's the dose, for example. That's one question we still need to answer. Can we reduce the dose of nanoparticles, the amount of nanoparticle material we give, and still get the same result? Do we need to potentially increase it? Do we need to wait longer periods of time before we do the imaging? So today in our study, we image at 24 hours and 72 hours post the injection. So it's at this stage, some of the things that we still need to learn are how best to optimize. What we know now is that we can inject the imaging agent. It seems to be safe and well tolerated. It is getting into the lymphatic system, but now we need to look at how best to optimize it. So when we go do multiple hundreds of patients to get clinical approval, we're actually doing it with the most optimized administration and protocol.
Thank you. Moving away from the trial, recent company announcements have had a high degree of ambiguity, and I'm presuming this is coming from our March trial update, especially, where more questions are raised than answered, and shareholders are left looking for hidden messages and attempting to read between the lines.
So I guess I would respond to that with, don't look for hidden messages, and we're not trying to be obtuse. We're very careful about reporting the facts, right? And we've said repeatedly, and what we've said in my chair remarks is that we're going to continue to disclose things that we consider to be material. And so I wouldn't try to read between the lines. If we report that we feel that in this example, that the evidence is suggesting that through the first 5 patients, we're seeing that it's safe and well tolerated, there's nothing more than that to it. And if we say that we are not yet ready to make a determination whether it's effective, it's because with an N of 5, we're not capable of making that conclusion. So I would encourage shareholders not to think we're trying to be coy. We're not trying to be cutesy. We're simply reporting what we can report that we see as being material information for the company.
Thanks, Bob. Another question from a shareholder. A statement and then a question. Why has the news flow dried up since November 2020? There have been few price-sensitive announcements in the last 18 months. Shareholders are of the belief that there was more going on in the background, which would lead to further news flow. Aside from the new facility, is there anything newsworthy on the horizon?
So I think I would say 2 parts to that. One is that, again, we are very clear that we make announcements when we think there's something material. So if the news flow feels like it's dried up, according to the question, it's because perhaps nothing material has happened. It doesn't mean nothing is going on. It doesn't mean that we're not undertaking R&D activities or that we haven't made progress, but it may not be material. And we're very careful not to just put out news just for the sake of putting out news. I would caution folks to look at no news as not being bad news. It just means that we haven't had anything material to report. We do include, as part of our communications, investor newsletter, which does provide insight and information as to activities that we have that may not be material by themselves, but we restrict our ASX announcements to those things according to ASX listing rules are material to the company. As for forthcoming, it's hard to predict when something material might actually happen, but we would say certainly that our progress with the development of the current clinical study, any meaningful progress for any of the other preclinical activity that we have, and or activity related to expanding the clinical study in jurisdictions outside Australia, we'll report them as they take place.
Thanks, Bob. Another question here. This might be more relevant for you, Di. I'll read a bit of the background. An independent non-executive chair is fundamental to good corporate governance, improving performance and accountability of the management team and reducing the potential for critical conflicts of interest. It is also a required feature for many institutions to invest in a company. Does the board of directors plan to appoint an independent chair in the future?
Thanks, Jeff. It's an important question. The company, through the Rem Nom Committee, actually conducts a separate chair performance review. So we go through an annual review of how the chair is executing that role as an independent chair as such, and then separately as a CEO. So we conduct 2 performance reviews. We are very mindful of our particular set of circumstances. But having said all of that, it is something that the company is looking to, reviewing, thinking of, and planning down the track. to move towards that model.
I think, Bob, it's fair to say it's not uncommon in the US.
It's not uncommon in the US, but again, we're an ASX listed company with a substantial shareholder base in Australia. And so if we don't look like the normal zebra that they're used to seeing, I think it's a fair question to ask. And as Di, as chair of the RemNom committee, has responded there, it's something that we do take a look at ongoing, and I'll be the first one to recognize if I don't think that it suits the company appropriately.
Yeah. Another question here, this is probably a better place for me to answer actually. Why haven't any directors bought shares on market recently or generally historically? It's a really good question, that, and I think some shareholders are the view that they like to see directors buying on market. Unfortunately, when directors buy on market, they can sell on market too, and that's not viewed as well. And other shareholders like to see directors actually not buying as many shares and being purely independent. So we really leave, there's no recommendations from management to any of our directors to be going and buying shares on market. That is purely their prerogative and for their decision. I guess another part to that question, do they see the shares as good value? We don't comment on the value of the shares and the share price, but what I can say is, just an outsider, having been involved in many listed companies, as a small company in the position we're in, we have a very high caliber board, and they wouldn't be giving us their time and effort and energy and commitment and putting their name to us if they didn't believe in the story. And I think that's more important than them actually going and buying 10,000 shares on market tomorrow to be seen to support the company in that way. They're doing far more than that, just being with us. So that's an answer to that question. The last question we have, and again, this is probably better for me to answer as well. Please explain the process of how the board, and this is from Mr. Ruben Pack. Thank you, Ruben. Please explain the process of how the board determines it is in compliance with its continuous disclosure obligations. Look, really good question. We have a board with some directors in Australia and directors in the US. We're an ASX listed company. We meet regularly as a board. We have our general counsel, Harry Kingsley, who's in the room today from K&L Gates. He attends all our meetings and is available on call. I myself have listed company experience spanning 14 or 15 years now, and keep up to date with all the continuous disclosure obligations of the ASX and the Corporations Act. We have Di and Ivanka both on listed company boards in Australia as well, to ensure we've got that good representation at a board level. Bob himself did the Australian Institute of Company Directors course not that long ago. And Mike and David and being, even though they're US based, have a lot of listed company experience in the US, doesn't translate directly to Australian laws, but is not that dissimilar in a general what's material, what's not material, how should we be disclosing information. And in addition to that, we take very seriously our disclosure obligations, and therefore have a disclosure committee that meets regularly whenever we need to in terms of discussing any potential announcements. So anything further from anyone else on that one? Or, I conclude that is the last of our questions, unless there's any more in the room.
Yeah, thanks. Just taking you, John Murray, by the way, just taking you back to the discussion about the trial that's currently being conducted. My observation is that the trial is actually quite detailed in terms of not only trying to determine the safety and efficacy, the safety of the nanoparticles, but also potentially provides some read on its ability to do what it's supposed to do. And so I do understand that currently you don't have sufficient statistical power to be able to say a lot about it. My question is whether whether you expect to have discussion of its efficacy. That's any information, pardon me, that's yielded from the trial when you get sufficient number of enrolments.
Yes. Short answer is yes, absolutely. So just taking a step back on that question, right? We could have done a typical phase 1 study of safety only, right? Safety and tolerability, taking non-breast cancer patients, naive patients, just see if we inject this imaging agent, does it hurt anybody, et cetera, right? But we wouldn't have learned whether or not we have the prospect of an imaging tool for detecting lymph nodal involvement. So that's what made the initial study a little bit more complicated than typical phase 1 studies. We had every expectation that it was likely to be safe and well tolerated. So just doing a phase 1 safety study wasn't going to move the needle for us very far. So the study, in fact, was designed to try to accomplish some measure of effectiveness and understanding while still demonstrating for ourselves that, in fact, our expectations of safety and tolerability were well founded. So that's exactly what we're trying to get to, is can we get to, and whether that N is 10 or 15, whatever we need in order to be comfortable that, in fact, what we're demonstrating is that we've designed a targeted magnetic particle that, if injected into a woman's breast tissue and picked up by the lymphatic system, is able to determine whether or not there's tumor involved in a lymph node. And so we expect that with somewhere between 10 and 15 patients, we're likely to be able to get that answer. And then we may still need to do, as I said earlier, dose optimization to make sure that we get, for the future product, that we really get that right. But what we would say today is that we have evidence that suggests it's moving in the right direction. If we felt that it wasn't working at all, we would stop the study. Our investigators would ask us to stop the study. There's no point in dosing patients if it's got no chance of working, right? Can I say today that, we'll say today with the N of 5 that we've reported interim results on, that everything is exactly the same? No, there is some variation across patients in what we are able to see, and that's expected. So that's why we need a bigger number. But the outcome of the study, when we achieve enough numbers of subjects, is in fact to allow us to make the decision, should we move this forward as an imaging agent because we've got evidence that it's going to work. Anything else, Di? You're very experienced in clinical studies, so.
All I'd add is it's situation law for clinical research. Very typically, you have to wait to the complete conclusion of a trial before you can speak, say anything, because the risk of misleading in a negative way as much as the positive way. So we do respectful of our shareholders that we're so anxious for these results as well, but are very mindful that early interpretations are fraught with danger. So we take an optimistic yet, as proactive approach as we can. And as Bob has said, when you start a trial, you start off with a number in mind, and you have to have a kind of a priori approach and then stand back from it as a very independent and well-run trial design. So as soon as we can, we will. Yeah. And we're looking forward to it very much.
And each new patient adds that little bit more information, but each new patient by itself is just another N of 1. And so we're very cautious. I mean, we get a lot of the investors that often ask, well, how many patients do you have now? When's the next patient, kind of thing? And it's on an individual basis, it's kind of like you look at a tree, but what we're waiting to see is the forest. And so hopefully, we'll get to that point where we can actually then take a look at the forest and say, what does this really look like? It's not helpful to report on an individual tree, so to speak.
A follow on. In hindsight, would it have been better to run a simple, regular phase 1 trial? Is it safe?
My answer to that is no. We could have potentially gotten through a larger number of patients that says, yep, safety and tolerability is good, but I still don't know. If you take a naive patient, for example, and you inject our imaging agent into them, and it goes into the lymph nodes, it'll tell us safety, tolerability, etc., but it won't tell us a thing about whether or not we could detect lymph nodal involvement. And that's the question we really wanted to de-risk, right? Because I don't want to go try to design a study for 200 or 300 patients if I don't have really good confidence that it's going to get us what we want. And just testing naive patients for safety would never give us an answer whether or not I have a low risk of being able to detect tumor involvement.
Bob, I think the other thing is the fact that we had a limited number to start with allowed us to really understand the kinetics of this and understand how long should we wait between further imaging exams, so we can go back and modify the protocol, be much more efficient going forward.
And also, if I can add,
please.
Love this question. But I think it's, the challenge and the beauty of the trial design that our nanoparticles are decorated with specific antibodies to a specific target. And that target's only presented on these type of cancer cells. So going halfway and saying we think we can see the pharmacokinetics and dynamics of how the nanoparticle passes through the body is good, as Mike has said, but the real prize here is that not only can you get through the body in a reasonable way and in a reasonable quantity, but actually attach to, prospective or suspected cancer cells in those lymph nodes. So that's what this is really about. So would it have been simpler? I think as a practical matter, probably not, because the patient has to go to the injection, has to be in the clinic, has to have MRI. There's still all these procedures. And but I take your point, because we are dealing with very, brave and terrifically admirable women who are undertaking this with a diagnosis of breast cancer. So they're all heroes in our eyes.
Yeah.
And but I understand the question.
Thank you.
Anything else? Yes, sir.
Yeah, great. Well, it seems that we're short on patients here in Australia for phase 1. Why can't we go to America?
So this gets to the why so many companies do in fact come to Australia for phase 1 type studies, and that is that the regulatory and clinical ethical environment here is very conducive to phase 1 studies. But the stringency in, for example, the United States, doing it with the FDA, or in the European Union, makes it much harder. So the material that we made, the imaging agent that we made, and the rules associated with getting the study done here made it easier for us to get the study done in a more timely manner and less expensively. If we were to turn around and try to do this in the United States, we would have to make a new lot of material that met FDA requirements, and then we'd still have to go through the regulatory approval process in the United States. So here you have an ethics committee that approves a study, and it's good for any institution that's within the purview of that ethics committee. In the United States, you have first to get the FDA's clearance, and then you got to get every institution to sign on with their own ethics review and their own what's known as an institutional review board. It's possible to do, but it's more time consuming and more expensive. And we've just felt that we could still achieve what we needed to achieve here in Australia, but we certainly have been impacted by the number of people being able to get into the hospital.
That would be the same as European.
It would be, yeah. European regulations are very similar to the United States, very stringent. Yeah, we have looked elsewhere. Admittedly, our team looked at a variety of different countries, and the problem is every country, bit of an exaggeration, most countries are different. So how you would do something in the United States, you'd have to do something different in China. You'd have to do something different in India. So we really did try to scour and see if what we've done here in Australia could be moved to another jurisdiction, but all the rules are different, and so it sort of meant starting all over again. And we just felt that we would spend a lot of time and a lot of money trying to get started elsewhere. If we just sat pat and stayed here and persevered in Australia, we'll still get to what we need to get to.
Yeah.
Okay. Any further questions? Come in.
Nothing else in the room? Yeah, nothing else online.
Thank you for your questions.